Welcome back to our course. Today we'll discuss about the lecture number 4, that is breeding for rice and maize. Here coming the lecture outline. So lecture outline is it is combined for both rice and maize crops. First, we'll discuss about the centers of origin, distribution of the species. Next is breeding objectives, conventional approaches, and innovative approaches that are nothing but conventional breeding methods and conventional and innovative, that is a biotechnology approaches. We'll discuss for both rice and maize crops. So this is the lecture outline for the rice and maize crop. First, we'll discuss about the rice crop. As we know, the rice is the most important staple food crop for the world, and whereas it is the most important staple food crop in India because half of the population is dependent upon this rice crop. So, the common name of it is a rice. The scientific name is Rhizos sativa. It is L. It is luminous. And next is chromosome number. It is having the chromosome around 2n equal to 24, and it belongs to Poaceae family. General, here we can discuss the molecular structural parts of the rice plant. Here you can see this is nothing but the culm. From this culm during the booting stage, the panicle were emerged from this boot leaf, and then after that the flowering will happen, and then the grains will form. These are nothing but the panicles. Here you can see the compact panicles. Uh, these are the grains. Um, these are the some of the structural parts of the rice. The rice is the most important staple food crop of the world. Around half of the population is dependent on this paddy crop, and the common it is called as rice or paddy. And the scientific name is Rhizos sativa L, and the chromosome number is 2n equal to 24, and it belongs to Poaceae family. And next, coming to introduction, as we know, the rice is the most important staple food crop of the world, and around 60% of the around this paddy will contribute around 60% to the world food basket, and mostly it is a warm season crop, and it is grown extensively in humid tropical and subtropical regions of the world. And next, coming to distribution, it is grown in China, India, Japan, Korea, and Southeastern Asia, and the adjacent islands of the Pacific, according around the 90% of the world rice production from these countries, that is in China, India, Japan, Korea, Southeastern Asia, and the adjacent islands of the Pacific. These countries are contributing around the 90% of the world rice production. And in India, the rice is the predominant crop, and it is the most staple food crop for the mostly for the eastern and southern parts of India. As we know, the rice is the oldest cultivated crop. Next, coming to the origin of the cultivated rice. Generally, the origin of the rice is classified into two classes. That first one is the polyphyletic origin, and the second one is monophyletic origin. First, coming to polyphyletic origin. Here, generally, in case of rice, we are having the two cultivated rice varieties. Two cultivated rice varieties. That is, first one is the Asian rice, and the second one is African rice. Asian rice is called as Oryza sativa, whereas African rice is called as Oryza glabrina. The origin of the Asian rice is South and Southwest Asia, whereas for African rice it is a tropical Africa. So, according to the polyphyletic origin, we'll discuss how this Asian rice and African rice were originated from this polyphyletic origin as well as the monophyletic origin. So the Asian rice is Oryza sativa and the African rice is Oryza glaberrima. And here origin for Asian rice is Southwest Asia, and for African rice it is tropical Africa. So now according to polyphyletic origin, how this cultivated rice were originated we'll discuss. According to polyphyletic origin, first this is the Oryza ruficogan. It is a wild perennial. From Oryza ruficogan, the wild annual, that is Oryza nivara, was obtained, and from Oryza nivara, that is a cultivated annual, that is Oryza sativa was originated. Again, this Oryza sativa is having three different forms, three different subspecies. That's Indica, Japonica, and Javanica. So the Indica, it is grown in temperate area and is originated in India. Next is Japonica, it is originated in the Japan, whereas it's mostly grown in the temperate conditions. And next is Javanica. Javanica is nothing but it is originated in the Indonesia. So it is also grown in the tropical countries, and this Javanica are called as bulu types. So this is how the Asian rice was originated from Oryza ruficorum. The Oryza nivara was obtained, and from Oryza nivara the Oryza sativa was obtained. This is the cultivated rice, that is the Asian rice. From the again this Asian rice is having three different species. That is Indica, Japonica, and Javanica. This Indica is originated in India, whereas Japonica it is in Japan, whereas Javanica it was originated in the Indonesia. So it is this indica types are grown in the tropical countries, whereas Japonica is type in temperate conditions, whereas Javanica were grown in the tropical conditions, whereas Javanica are also called as bulu types. So this is how the Asian rice was originated, and these are the three subspecies of the Oryza sativa. Then coming to the African rice, according to the polyphyletic reason, how African rice was originated. The first from this Oryza, this is the Oryza longus caminata. From Oryza longus caminata, the Oryza burti was obtained, and from Oryza burti, the Oryza glaberi were obtained. So this is how the African rice was originated. Asian rice, so this is nothing but polyphyletic. Polyphyletic origin is nothing but from different species. This cultivated rice were originated. So this is called as polyphyletic origin. Here you can see from different species. Here we got the Asian rice, and here also from the different species we got the African rice. So this is called as polyphyletic origin. And next is monophyletic origin. Monophyletic origin is nothing but here here the African rice, whereas Asian rice were original. These two cultivated rice were originated from the common parent or common ancestor, that is nothing but Oryza perennis. So here the cultivated rice is originated only from the one common parent or common ancestor. So it is called as monophyletic origin. So here the origin is grouped into two classes, that is polyphyletic origin and monophyletic origin. In this we'll discuss how this Asian rice and African rice were originated from different species, that is a polyphyletic, and next is monophyletic origin, and from the single parent this cultivated rice was originated, so it is called as polyphyletic origin. Next, coming to important objective, that is breeding objectives. Here we are having so many breeding objectives in case of rice. So we'll discuss one by one. The first one is the high yield potential. In every breeding program, the yield is the most important character, which will contribute which will contribute more towards the breeding program. So generally, the yield is a complex character because it is governed by many genes. So it is having the complex character. To in case of rice, to get the higher yields, the plant should have a short stature, and as well as it should have thick and stiff skull, and it should have compact panicle, and it should have short, narrow, and erect leaves to utilize proper solar radiation, and the flag leaf should not be more than 100. So these are some of the ideal plant types. The character These are the ideal plant type should have this type of characters like short stature, thick and stiff skull, and the compact panicle, and the short and narrow erect leaves. And next is the leaf angle. The flag leaf angle should not be more than 400. So these are the characters which will more contribute towards the yield in case of rice. Next is early maturity. Early maturity is nothing but generally if the duration of the crop is less, we can go for multiple cropping or different or crop rotation. So because of this because of the short duration, we can overcome the terminal drought, and we can also escape from the pest and disease attack. So this is the main use of the early maturity. The main thing is because of the short duration, within a year we can go for 4 to 5 seasons we can raise the crop, and it also helps to overcome the drought in rain-fed areas, and it also helps to escape from the pest and diseases. That is the early maturity. In case of rice, if the early maturity is around 1 or 5 days, when the duration is still reduced around below 1 or 5 days, automatically the yield will be reduced. So we have to take care while reducing the duration of the crop. So we are having here the rice varieties, that is CR666, Axi, and Co41 are the varieties which are having less than 100 days duration. Next is resistant to lodging and shattering. Same as like brain wheel, here the lodging and shattering also is the complex characters. They are also governed by the polygenes. Polygenes are nothing but these are the here the characters are governed by many genes and which are having the more influence of the environment. So that are nothing but polygenes. So because of that, this lodging and shattering is having complex character. Generally, the non-lodging line should have short stature and thick, strong culm and sharp internode, and the leaf sheet should tightly encircle the culm. So, generally, if the uh, plant height is dwarf and it should then if the heavy winds comes also the plant will not lodge. And if the plant is dwarf and the stem thickness is more, that is thickness is more, and then it is having the short internodes, then also so that the type of lines are also having the more resistance to lodging. And so these are the four characters where the non-lodging line should have, that is short stature, thick strong culm, short internode, and the leaf sheath should encircle the culm tightly. So these are the four characters which will contribute to the non-lodging lines. And next is grain shattering. This grain shattering is it is also the complex character like lodging. So generally the wild rice is having the more grain shattering. So while selecting this uh, wild rice, we have to take care because if this thing is transmitted to the cultivated species, cultivated lines, again this cultivated lines will have the same grain shattering problem. So we have to take care while crossing, while hybridization. This is about the resistant to lodging and shattering. And next is resistant to cold temperature. Generally, this Kumbhan Valley and Budalkar Taluk of Nilgiris are more suitable to this cold tolerance. For this, the Japonica rice varieties are more cold tolerant. So here you can see the examples for this. That is MDU2, that is a cold tolerant variety, which is a cross between 425 and IR8 rice, and next is resistant to salinity and alkalinity. Mostly in some areas of Raiki and the Dharmapur districts of Tamil Nadu, we can face this salinity and alkalinity problems. For that also we are having some of the rice varieties. For old varieties are SR26B, Get2 and Dasal, and the latest are the Co43. That is, it is a cross between Dasal into IR20, and next is ADT35 and TRY1 and TRY2. So these are the some of the rice varieties which are having resistance to salinity and alkalinity. And next is improved grain quality. Generally, after the major breeding objective in rice is it is paddy. Next is after high yield, the most important breeding objective is quality. So generally, the rice cultivars are classified based on the size, shape, and the texture of the grains. When these rice cultivars are classified into like based on the length, shape, and texture, the rice cultivars are classified. After that, this classification was given by Food and Agriculture Organization. So, based on the after culling and milling, we'll take this based on the length and shape, we'll divide the rice cultivars for the length. If the if the grain size is the grain length is greater than 7 mm, it is considered as the extra long variety. If the grain is between 6 to 7 mm length, it is called as long variety or long grain type. And next is if the if the grain length is 5 to 5.99 mm, it is considered as a medium type. And next is if the grain is length is below 5 mm, it is called as short grain. So based on the length, they are classified into four types. That is extra long, long, medium, and short. For extra long, the length should be greater than 7 mm, and for long it is 6 to 7 mm, and for medium it is 5 to 5.99 mm, and for short it is below 5 mm. And next, based on the shape, here we'll calculate the length by breadth ratio. Generally, suppose we are having one sample. From a sample after hulling and milling, we'll take the random of 10 to 20 grains and we'll do the mean values. First, we'll do the mean values of the length. First, we'll calculate the length, and next we'll take the breadth value of all the 10 grains, and then we'll calculate the length of the mean length of the length by mean length of the breadth. We'll take, and then we'll classify them into slender, medium, and bold. If the L by B ratio is greater than 3, it is called as slender type, slender grain type. If the L by B ratio is between 2 to 3, it is called as medium type. If the L by B ratio is between 2 to 2.39, they are called as gold grain type. So, this Food and Agriculture Organization have classified the rice cultivars based on the size, shape, and texture. Based on the length, they are divided into four types, and based on the shape, they are divided into three types. Next is breeding for alternate source of dwarfing gene. Generally, nowadays all the presently cultivated cultivars are having the dwarfing gene, that is DGO gene. This DGO gene, mostly we are using this dwarfing gene in case of all the present cultivated cultivars. So if this gene becomes susceptible to some new pest or diseases, the entire breeding program will collapse. So we have to develop some of the alternate source of dwarfing gene in the rice. So this can be done. This alternate source we can develop through using some of the conventional and non-conventional breeding techniques. And next is breeding for pests and diseases. Generally, the major pests in the paddy are stem borer, brown plant hopper, rice gall milt. So these are the three important pests which are present in the rice crop. And next is the major diseases. Major diseases are the blast, and next is brown spot, and the bacterial leaf blight, and Tungo virus. These are the four important diseases which are uh, present in this paddy. So for this stem borer, the donors are present. That is TKM6 and IR20 against this stem borer. And for disease, that is blast resistant genes, we are having the different donors. That is IR20, it is a medium duration, and 437, it is a short duration, and 425, it is a long duration. And here the grass stem virus, which further the we have the donor from the horizon Ivara. So this is about the breeding for pest and disease resistance. And next, so these are the 8 important breeding objectives in case of rice. And next, coming to the breeding techniques, that includes both conventional as well as innovative breeding methods. First, we will discuss about the introduction breeding method. Generally, in rice, all the introduced rice varieties are from the IRRI. IRRI is nothing but International Rice Research Institute, which is located in the Philippines. Generally, all introduced rice varieties from Philippines from IR8 to IR27 all are introduced, and the examples for them are the Basmati was introduced from the Punjab, and Ponni, that is a Mahasuri from Malaysia, and CR1009, that is Ponnamani from Orissa. So all these are the introduced rice varieties through introduction method. And next is pure line selection. So pure line selection, that is 49, it is a short duration variety was developed. And next is 432, that is Tiruchengodu Samba, it is a medium duration variety. And next is 490, that is Chengalpattu Sirmani, that is a long duration variety. Through pure line selection, they have developed the three different types of varieties in case of rice. And next is mutation breeding. Generally, the mutation breeding is mostly used in the rice. That is, we'll be having two different types of mutations. That is spontaneous mutation, and next is induced mutation. Through spontaneous mutation, they have developed the two mutants, that is JEP24 and ADT41. This JEP24 was developed from Arthur Kichlisamba. It is known as Konnamani, which is a fine grain and which is having a good quality rice. So from this source, the JEP24 mutant was developed. And next is ADT41. This is the ADT41 mutant this is developed from the dwarf mutant of Basmati 370. So these are the two spontaneous mutation induced mutants in case of rice. And next is induced mutation. Through different chemical methods or through X-rays or gamma rays, we can induce the mutation. So Jagannath rice was introduced from Orissa, which is a semi-dwarf variety. And next is the Prabhavati. Prabhavati is a short duration variety, as well as here it is a gamma irradiated mutant. And next coming to heterosis breeding. Heterosis breeding is nothing. Generally, first heterosis is nothing, but it is the here the F1 is superior over its both the parents is nothing but heterosis. This heterosis the F1 will be superior in case of yield, yield, yield or vegetable growth anything. If the F1 is found superior to both the parents, it is called as heterosis. The heterosis breeding, they have developed some of three hybrids that is 4RH1, 4RH2 and ADT-RH1. So this is about the heterosis breeding. And next coming to how the hybrid rice is developed. Generally as we know, the rice is the strictly self-pollinated crop. So to develop the commercial hybrids, we are using the male sterility system. This male sterility system is nothing but it is characterized by the non-functional pollen grains. So here the rice panicles will be unable to set the seeds through selfing. That is nothing but male sterility system. Generally, the male sterile lines are used as a female parent of the hybrid. So here we are having the genetic and non-genetic male sterility system to develop the rice hybrids. The first one is cytoplasmic genetic male sterility, and next is environmental sensitive genetic male sterility, and the chemical induced male sterility. So these are the three different types of male sterility systems used in the rice. So here we'll discuss about the two-line system and three-line system. Three-line system is nothing but CGMS system. So here we can discuss in generally in CGMS system we are having three different types of lines. A line, this is the A line, and next is B line, and next is the R line we will be having. Generally for hybrid seed production, we require three lines, that is A line, B line and R line. First, this A line is called as male sterile line or male fertile line. Generally, in A line, the nucleus will be here. The cytoplasm will be sterile in case of A line. Next is hybrid seed production, and next is B line. The B line is called as maintainer line. It is also male fertile line. Here you can see the cytoplasm is fertile. So this A line is crossed with the B line. We'll get the male sterile A line. So here also the male sterile line is here the cytoplasm is also sterile. You can see. So next for the hybrid seed production, we have to cross this male sterile A line with R line. Here the R line is nothing but restorer line, which is used to restore the fertility of A line. So here the cytoplasm is fertile in case of R line. So automatically we'll get the fertile F1 hybrid. So this is about the CGMS system which is used in the rice. This is here also you can see here this is the CMS line, that is A line, and this is the B line. Then we'll cross this A line and B line, we'll get the that is a CMS line. Again this CMS line is a male sterile line, so it is crossed again with the restorer line. When we'll cross with the restorer line, we'll get the F1 hybrid. So here A line, B line, and this R line. So this is nothing but three-line system which is widely used in the hybrid seed production in rice. And next coming to two-line system. In two-line system, this EGMS is nothing environmentally sensitive genetic male sterility. Environment sensitive genetic male sterility is nothing but generally, we will be having photosensitive genetic male sterility and thermal insensitive genetic male sterility. So those lines will be crossed with the restorer lines and we'll get the F1 hybrid. So based on the environment, the lines will convert into fertile or they will convert into the sterile. That is nothing but environment sensitive genetic male sterility. So this three-line and two-line system are uh, widely used in the rice hybrid seed production, and one line system is also used, that is through apomixis. And next coming to the next biotechnological approach is the mark resisted selection. Mark resisted selection is nothing but it is an indirect selection for the plants. Generally we'll use the different types of markers we'll use, and those markers which are linked tightly to the desirable traits. Based on that we can identify the positive plants by using this DNA marker. So that is nothing but mark resisted selection. And next is institute. Generally, these are the three institutes which are working for rice. That is International Rice Research Institute, which is located in the Philippines. All introduced IR 8 to IR 72. All the introduced varieties are from the Philippines, that is International Rice Research Institute. And next is the at national level, these are the two institutes which are working. That is first is the Indian Institute of Rice Research. Formerly, it was called as Directorate of Rice Research, which was located in the Hyderabad, Telangana. And next is the National Rice Research Institute, that is the NRRI. It was located in the Karnataka and Odisha. From this NRRI, they have developed some of the biofortified rice varieties. There are two biofortified rice varieties they have developed, CR Dhan 310 and CR Dhan 311. This CR Dhan 310 is it is having a higher amount of the protein, that is around 10.1% of the protein content. And next is CR Dhan 311. It is having 10.2% of the protein content. So these are the two biofortified CR Dhan 310 and CR Dhan 311. These are the two biofortified, that is more amount of the protein they have developed from this NRI Institute, Cuttack and Orissa. So this is about the rice crop. Next, coming to the maize. Maize, as we know, the maize among all the cereals, the maize will have the more amount, will give the more productivity is seen in case of maize crop. So it is called as queen of cereals. But a king of cereals is the wheat crop. Here the scientific name of maize it is a Zea maize, and it is a diploid species. So it is having the chromosome number 20. Generally, the productivity is more among the squirrels, so it is called as queen of squirrels, and it belongs to Poaceae or Gramineae family. And the origin of maize it is in Mexico or Central America. Generally, out of all crops, most of the research was done on this maize crop, and generally among all cereals, it will give the more productivity. So, and generally, this maize is fed to the poultry. Around 61% it is used for the poultry feed. So here we can discuss the structural parts of the maize plant. Generally, the maize is a monoecious plant. What is a monoecious condition means? Monoecious condition is nothing but the presence of both male inflorescence and the female inflorescence on the same plant at different positions is nothing but monoecious condition. So at the top of the plant we will be having the male inflorescence, that is called as tassel, and next is at the axillary position we will be having the female inflorescence. It is called cob, and here this cob is having the silk like a thread-like structures, that is nothing but silk. So these are the structural parts of the maize. And next, coming to origin and distribution, generally it is grown in USA, China, Canada, and South Asian countries. And this maize is having uh, two progeny, that is Gia tunicata and Gia teoscinate. And next is wild relatives. Generally, just now we discussed this Gia genus is having the male terminal inflorescence with the paired staminate spikelet and the lateral female inflorescence with singular paired spikelet, spikelet. Next is coming to the classification of the maize kernel. Just now we discussed the cultivated species of the maize is diploid. So this it is having two close relatives, that is the gamma grass and teoscinate. Gamma grass is the tripsacum, whereas the teoscinate it is the Zea mexicana. These are like grass type. Grass type. These are the close relatives to the cultivated maize species. So based on the endosperm, the maize is divided, the corn or the maize is divided into seven different types by Sutherland in 1899. So here we are having the seven different types of corns. Dent corn, the scientific name is Zea maize var identata, and next is flint corn, that is a Zea maize var indurata, and next is sweet corn, this is a Zea maize saccharata, and next is flour corn, this is a Zea maize amelaceae, and next is popcorn. It is the scientific name is Zeamis everta. And the popcorn, the Zeamis tunicate, and the waxycorn, it is a Zeamis keratina. So so each and every corn we will be having generally some endosperm characteristics they are having. Here the sweet corn, generally the sweet corn is harvested at the 15 day at the before physiological maturity we can harvest, and we can eat this sweet corn, and which is having more sugar content. And next is flour corn. Flour corn is used in different types of different types of fry making. And next is popcorn. As we every know, most of the this endosperm is very hard, and we can use this for making the popcorn. And next is popcorn. Popcorn is the primitive corn which is not widely used. And denticon and flintcorn, these are not much used. So based on the characteristics of endosperm, they are classified into 7 different types. Next, coming to the breeding objectives. In every breeding program and in every crop, the yield is the most important character which was contributing towards. So here the yield is also a complex character which was governed by the polygenes. So this ideal video type for maize was given by the Mock and Pierce. So generally for maize, the varietal hybridization is not popular because as it is difficult in getting the superior segregation. So this yield is a complex character. It is governed by polygenes. So to get the higher yield, the more generally they have developed the single cross hybrids and double cross hybrids. In single cross hybrid, compared to single cross hybrids, the double cross hybrids will be having the more amount of the yield when compared to the single cross hybrids. And next is coming to the um, breeding for biotic stress resistant. That is generally the major pest in case of maize is shoot fly and heliotis, which are Mexican varieties, which, which are to which the Mexican varieties are resistant. So these are the shoot fly and heliotis are the two major pests which was present in the maize crop. Next is the major diseases, the downy mildew and leaf blight. This downy mildew and leaf blight are the major diseases in case of maize because this we are having the texture cytoplasm, it became susceptible to the downy mildew. And next is here the co19, co-H2 are and co-H2 are resistance, and the Taiwan lion is resistance to the downwind. Generally, this is a CMS, that is a cytoplasmic male line, that is a Texas. It is became susceptible to the helminthosporium, but this CMS-C and CMS-S cytoplasmic male lines are resistant. Only this Texas line was susceptible to the helminthosporium. These two CMS lines are not susceptible. They are resistant to the and next is protein content. Generally, the main amino acids present in the maize are lysine and tryptophan, and the protein of the endosperm in maize is called as gene. But this is having the lesser amount of the lysine and tryptophan. So because of that, the nutrition is unbalanced. It is unbalanced. So they have developed the quality protein maize. That is QPM. QPM is nothing but it is a variety which was developed with higher amount of lysine and tryptophan, and generally the embryo contains around 20% of the protein content, which have higher amino acid content. And the best examples for the quality protein is Shakti, Ratana, and Protina. So these are the uh, three varieties which were developed, which are quality protein maize with a higher amount of lysine and tryptophan content. And next is oil content. Generally, the maize is having linoleic acid and linolenic acid, and the embryo contains around 12 to 15% of the oil content. The protein content is called as bean, and around the embryo is having 20% of the protein. And the quality protein is generally the normal maize is having lesser amount of lysine and tryptophan. To get the balanced nutrition, they have developed the quality protein maize with higher amount of lysine and tryptophan. For that, the examples are Shakti, Ratana, and Protina. And next is oil content. Here, the maize is also having the embryo is having around 12 to 15% of the oil content. It have linoleic and linolenic acid, unsaturated fatty acids. And next is high yielding baby corn. The scientific name of the baby corn, it is a Giamma saccharata. Generally, these green crops are eaten as a salad. For that, the best variety of the baby corn is Co-BC1. And next, coming to breeding methods. Generally, the Portuguese was first introduced the maize into India, where the Mexican lives, and the initial varieties all are introduced, like the Sikkim Primitive 1 and Sikkim Primitive 2. These are the two introduced lines varieties. And next is mass selection. Before 1945, mass selection was only a method, but some mass selected varieties are KT1 in UP and RAS1 in Rajasthan. And next is year to row method. This year to row method and modified year to row method are mostly used in the maize crop. Generally, year to row method. This year to row method was proposed by Hopkins. This is the exclusive method for the oil and protein improvement. Generally, this method is suitable for the characters which are having the high irritability, like oil and protein content, but not for yield. It is mostly used to develop the oil content and protein content, but not for the yield. So this was developed by the Hopkins, that is the year to row method. And next is modified year to row method. This was proposed by the Longquist. Longest here you can see detailing year to row method. Year to row method, here we will be having the three cycles, that is the first selection cycle, second selection cycle, and third selection cycle. In the first year, the plants are grown and the plants are selected based on the phenotype, and then the open pollinated seed were from each the plant will be harvested separately. And after that, in the second year, whatever the harvested seed are grown in the progeny rows from the selected plants, and then from that the here the progeny rows are grown from the selected plant, and the superior progenies are identified and selected, and they are allowed for open pollination, and after that the seeds are harvested separately. Again, in the third year, the same thing was repeated whatever the done in the second year, and then this may be repeated one or more times, and then afterwards it will be done for the yield trials. This is nothing but year to row method. Next is hybridization and selection. This is generally the first hybrids of maize are Ganga 101, Deccan and Ranjit. These all are double cross hybrids. And next is heterosis breeding. Heterosis breeding is nothing but, as we discussed here, the F1 will be superior over its parent. That is nothing but heterosis. So here they have crossed some of the different lines. That is Indian into Indian they crossed. They got about 24 to 43% increase in the yield. Next, Indian into US length they crossed. They got about 58% increase in the yield. And next is Indian into Caribbean flint they crossed. They got 47 to 54% increase in the yield. So in the heterosis breeding, we are having different types of hybrids in case of maize. That is single cross hybrid, three-way cross hybrid, double cross hybrid, and double top cross hybrid. So here single cross hybrid is nothing but it is a cross between two inbred lines. It is nothing but A into B. It is a cross between two inbred lines is nothing but single cross hybrid. And next is three-way cross hybrid. Three-way cross hybrid is nothing but it is a cross between A into B into C, or it is a cross between single cross into inbred line. So the examples for this is Ganga 5 and Trishulata. And next is double cross hybrid. హలో మేడం, తీసుకొచ్చిర్రా? సరే మేడం, నేను వస్తాను. నేను ఇటు వేరే ప్లేస్ లో ఉన్నా, వస్తా. లేకపోతే సుబ్బారెడ్డి సార్ రాకుంది కదా మేడం, అందులో పెట్టండి. నేను నేను వస్తాను. లేకపోతే మీ బ్యాగ్ లో పెట్టేసుకోండి, మీది ఉంటది కదా ర్యాక్. సరే మేడం, ఓకే. Next is double cross hybrid. Double cross hybrid is nothing but it is a cross between two single crosses, or it is a cross between four inbred lines. That is A into B, that is crossed with C into D. That is nothing but double cross hybrid, that is 4H3. And next is double top cross hybrid. It includes a white kernel hybrid, Ganga Safed 2, High Stars, and Ganga 4. These are the some of the hybrids which are developed that are nothing but double cross hybrid. Next is population improvement. Population improvement method is nothing but it is a recurrent selection technique which was initiated by Darwin in 1963 in maize. The main aim of this population improvement is to isolate the superior inbreds from their population for hybrid and synthetic variety production. Next important points on this maize is, as we discussed earlier, it is a monoecious plant because both male and female inflorescence is present on the same plant. same plant on the different places is nothing but monoecious condition, and it shows a protandry nature. Protandry nature is nothing but the stamens mature earlier than the pistils. So that is nothing but protandry condition. So here the tassel mature earlier than the pod. So it is called as protandry condition. That's why it shows the, that's why it is a cross-pollinated crop. And next, the male inflorescence is present in the top, that is at terminal position, so it is called as tassel, and the female inflorescence is present in the lateral axil, that is called as pod. So this is about the inflorescence, and next we will discuss about the institutes and the programs in the maize crop. That is the first major institute which is working on this maize is CIMMET, that is International Maize and Wheat Improvement Center, which is located at Mexico. And next is IIMR, that is Indian Institute of Maize Research. And next is Maize Research Center, which is located in the Hyderabad. And generally, the ICRIP is nothing but All India Coordinated Research Improvement Project, which was first started on the maize crop in 1957. So this is about the institute and programs. And coming to the summary, as we know, the rice is the world's leading staple food crop, which was improved through the breeding for the yield, early maturity, stress tolerance, and habits using the maize sterility system. And whereas the maize is a highly cross-pollinated, it is a versatile cereal with breeding focused on the hybrids and quality protein maize and recurrent selection program. So this is about the breeding for the rice and maize crop. Thank you.